afternoon, everyone. I'm Mengjun Hu. I'm a small fruit pathologist, University of Maryland, same as the last speaker, Dr. Faku. So located in College Park, and I work with small fruits as well as wine grapes. So I guess today I'm just going to talk about fungicide resistance. So first of all, how many of you are using fungicides? Just wondering, okay. And how many of you are concerned about fungicide resistance? Good. All right, well, hopefully this will be useful to you. Now I will go through this quickly, since many of you are expert on this, why are we using pesticides, right? Of course, as you know, 3 factors are required for disease infections. We got to have a susceptible host, conducive environment, and the presence of pathogen. Now, in the Mid-Atlantic, as you know, our weather conditions are warm and wet, right, during growing seasons, and that considered inducive environmental conditions for fungal infections and bacterial infections as well. And pathogens, many of those pathogens are airborne, so they can travel for a long distance, and there is no shortage of pathogens really in our environment. And host, and I work with small fruits, wine grapes. Those are perishable, high sugar content, and many of them are susceptible to fungal pathogens, so diseases, right? That's why we're using pesticides, including fungicides, to control the disease. During the establishment and the development of a crop, and sometimes to improve the storage life, right? And the quality of harvested plants and produce. So, okay, just a little bit more about fungicides. In general, there are 2 types of fungicides, as you know. One is what we call multi-site fungicides or protectants. Sometimes you can also call them contact fungicides, right? And there are pros and cons of each fungicide type. And the pros of multi-site fungicides they are less expensive, right? Most of them are old chemistry. They're less expensive. They have a broader spectrum of activity, but they're also less prone to resistance development. And concerns with those multi-site fungicides many of them are potentially phytotoxic. So you got to be careful with using multi-site fungicides. Sometimes if they can result in crop injuries, right, if used improperly. And they have a preventative activity only. So they do not have post-infection efficacy, and they will not protect the new growth, right? And oftentimes they're also prone to wash off by rainfalls and sunlight degradation as well. And then typically our growers consider, we need to reapply those multi-site fungicides more often. Okay, single-site fungicides, the pros with single-site fungicides are some of them have a kickback activity, or you call creative activity, right? And they do not have to be applied as frequently as multi-site fungicides. And because the way that it works is that it attacks a specific site in a metabolic pathway, right? So which makes them less toxic in general. So they have a favorable toxicity profile, which are favored in our IPM programs, right? But the concerns are, is because of that site specificity, the improved site specificity, they're much more prone to resistance development. So organisms, they're more easily to overcome fungicides, single-site fungicides, having resistance issues. And also they're more expensive, right? Okay, now I talk about multi-site fungicides, single-site fungicides, but how we distinguish those, how we identify them. If you look at fungicide label, pay attention to group fungicide, and that tells you whether they're multi-site or single-site fungicide. M, for example, Captain, that's a multi-site fungicide, and it has a group code M4. Now M stands for multi-site, right? And the other one, Redmill Gold, and that's a single-site fungicide. If you pay attention again to the group code, that's number 4 without M, so that indicates they're single-site fungicides, okay? All right, now I'll talk about fungicide. Now we're talking about fungicide resistance. Again, wine grapes, small fruits, and the tree fruits, In our environment, require a lot of fungicide applications, and wine grapes, it's typical for us to spray anywhere between 12 to 16 applications per season, and apple peach up to 20 sprays per season. Correct me if I'm wrong. Strawberry 6 to 10, brambles, depending on cultivars, can be up to 12 applications per season. So there are, the fungicides are being used very frequently, right? That's one factor that really speed up the resistance selection. And another factor is that we do not really have a lot of chemical classes of fungicide that we can use, that we can rotate with, right? FRAC 1, 3, 11, and those 4 chemical groups together, they are accounted for greater than 70% market, so overall usage. And you can see that we have limited chemical groups that we can use. This figure shows a relative importance of different fungicide groups used as a foliar sprays to control fungal cereal diseases in the United Kingdom. And you can see out of those different chemical classes of fungicides, QI fungicides, DMI fungicide, MBC fungicide, those are the majority, only 3 groups, right? Those are the predominant. And again, just shows you that there is a limited group of fungicides that we use. And that's another factor driving the development of fungicide resistance. So, it's not really a new problem. And this issue was noted back in 70s. That's right around the time, right after the time when single-site fungicides were introduced to the market. And then 40, 50 years later, it's still an ongoing issue and a big issue, right? Part of the reason, I think, is that, again, single-sites are favored in our spray programs. And then among pathogens developing fungicide resistance, botrytis certainly is deserves a sort of a spotlight due to tremendous ability to develop resistance to many chemical classes of fungicide that we currently use for botrytis control, right? And you can see this figure shows you the increase of fungicide resistance over time. Back to 1977, there was a little fungicide resistance in plant pathogens, and over time, you can see there is an increase of resistance, right? And this is not really limited to plant pathogens, but the same for human pathogens as well, right? And the antibiotic resistance, it's basically the same because we're using them more frequently because the novel medicine drugs that we have, they have improved site specificity, give you better toxicity profile, but meanwhile, they're much more prone to resistance development. Okay, now how exactly fungicide resistance is developed? You may be wondering, right? So basically, let's say you have an initial population where most of your individuals are sensitive, right? But over time, when you apply fungicides, single-site fungicides, sensitive individuals disappear, and then resistant individuals got selected by fungicide applications so that they are increasingly appear. And in a different scenario, you still have your initial population where most of your individuals are sensitive, right? And then depending on the phenotypes, resistant phenotypes and the mechanism of fungicide resistance, the individuals with intermediate and a high degree of resistance increasingly appear as fungicide application continues. So anyway, it's a overall, basically this is a resistance development is a selection process with the fungicide as the selecting agent, okay? Now, you may be wondering about the mechanisms of fungicide resistance. What confers fungicide resistance? What's the underlying mechanism? In general, there are 3 major types of mechanisms responsible for fungicide resistance. The first one is mutations in the targeted gene. As I mentioned earlier, that single-site fungicide, they attacked a specific metabolic site, right? And that site, what we call targeted gene for that fungicide. So the mutation in that target gene can confirm resistance to that fungicide, and oftentimes confers high level of resistance. And because of that site specificity, there is no cross resistance, okay? This mechanism typically does not confer cross resistance with other unrelated chemical classes fungicides. An example is G143A. That's a mutation in cytochrome B gene, which is a target gene of strobilurin fungicide 11. Another mechanism is overexpression of the target, and the increased efflux pump activities can also be responsible for fungicide resistance. There are proteins, genes in organisms that are that we call transporter proteins, and what they do is that they can pump out toxic substrates from cells in order for organisms to better survive and reproduce. And this type of mechanism, it can confer cross resistance. For example, increased ATRB activity. ATRB is a transporter protein and confers multi-site fungicide resistance. So anyway, now we talk about the issue, the fungicide resistance issue. Now we're talking about, let's talk about strategy, how we better manage fungicide resistance. Alternation and a mixture of fungicides with different modes of action. I think those are general resistance management strategies. Would you agree, right? So we're told to rotate and a mix of fungicides with different modes of action in order to better manage fungicide resistance. So alternation, basically you apply fungicides in alternation. And mixture is that the fungicides are applied in mixture. And a lot of products that we currently have, they're actually pre-mixed, right? They have different active ingredients, belong to different fructose, to different groups. And that basically is to hopefully better manage fungicide resistance by having those products with, pre-mixture. I'll come back to that later, but I guess maybe 10 years ago, 9 years ago, sometime around that time, I was involved in a resistance monitoring program to detect for fungicide resistance in botrytis from strawberry fields from East Coast. I've shown this picture many times. But again, the x-axis is different active ingredients that we tested, different fungicides, and the y-axis shows you the frequency of resistant isolates, right? So over time, you can say my point is that there is the increase of fungicide resistance to different chemical classes of fungicides commonly used for strawberry production, okay? And then, in addition to monitoring resistance to different individual fungicides, we also look at multi-fungicide resistance in the same isolates. And the x-axis that shows CCR, 0, 1, 2, 3, CCR, whatever. CCR stands for chemical class resistance. So 0 CCR means that the isolates are completely sensitive to fungicides we tested. Whereas 7 CCR means isolates are super isolates. They're resistant to all chemical classes fungicides that we tested. Okay, so just want to show you, over time, we observed that there is a shift towards multi-fungicide resistance. And we are kind of wondering, why, right? Because the part of the monitoring program was to better help growers manage fungicide resistance. And this is kind of surprising. And, during the monitoring program, we provide the test results to growers who participated in this program, basically to ask them to avoid using fungicides that have resistance issues based on the results, but also use fungicides that did not really have much resistance issues based on location-specific resistant monitoring test. But still, there is an increase, right? And then we think that the chemical class alternation may not be sustainable for multi-fungicide resistance management. I'll try to explain with this slide. In the first scenario, right, where you have a population, most of your individuals are sensitive, but you have a few individuals that are either resistant to fungicide A or fungicide B. And there is one individual that is resistant to both A and B simultaneously, okay? And once you spray fungicide A, of course, you're selecting for resistant A phenotype, right? You're selecting for individuals that are resistant to fungicide A. And over time, you just stick with fungicide A or spray A, spray A, spray A without rotation, okay? And in the end, it's easy to presume, to predict that in the end, your population where most of your individuals are resistant to fungicide A only, right? But in a different scenario, again, it's the same population, start with the same population. If you start with A, and now we're seeing some resistance issues with fungicide A, now we're gonna rotate with fungicide B, right? And then what does that rotation do? That rotation will, we think, speed up the selection for that dual resistant phenotype, right? So in the end, at the end, the individuals are resistant to both fungicide A and B will be accelerated. The selection for that dual resistant phenotype will speed up, okay? And so we think that the more fractals, more chemical groups are involved in a spray program, you're likely speed up the selection for multifungicide resistant phenotypes as well. Okay, so the alternation of fungicide may not be a sustainable way for managing multifungicide resistance, especially when you have initial populations where those dual resistant, multifungicide resistant phenotypes have already existed, okay? Now, it's also noteworthy that there are botrytis isolates that are simultaneously resistant to all chemical classes of fungicide that we tested. And I talk a little bit about mechanisms of fungicide resistance. In case you're wondering about that, you can say those two isolates, isolate A and isolate B, those contain different mutations in targeted genes that are responsible for different fungicides resistance. For example, boscalid, that's FRAC7, right? The targeted gene of that fungicide is SDHB gene, and the mutation in that gene, either H272R or 272R, 272Y, and they can confer resistance to boscalid, and so on. There are other targeted genes and mutations. Those genes can be responsible for resistance. So what I'm saying is that the multifungicide resistance mechanism in botrytis is an accumulation of mutations in targeted genes, okay? All right, well, I'm going to, let's delve more into fungicide resistance management. First of all, what's FENIS? What's FENIS? It's like sort of a healthy status, right? But in biology, fitness refers to organisms' ability to survive and reproduce in a particular environment. So we think that for isolates to become resistant, it gains resistance, but the resistance may also come with a price, with a cost, and that cost may be related to fitness, right? So it's sort of a little offset. So there is a possibility that fitness cost may be associated with some resistant phenotypes. Now the idea, but we really haven't really understood fitness cost associated with resistance that much, not in depth. But we think there is an opportunity to better understand fitness cost that would allow us to better or to improve our resistance management strategies. I'll give you an example. So if there is a fitness cost, right, that means the resistant strain is at a selective disadvantage when the target fungicide is not used, right? So over time, it will lead to a decrease in the fraction of the resistant population, in the resistant pathogen population. So in that case, alternation makes perfectly sense because that alternation we extend the spray interval of fungicide A, right? With that extension, we give sensitive isolates more time to take over the population, to outcompete resistant phenotypes, taking advantage of that fairness cost in resistant strains, right? But if there is no fairness cost, without a fairness cost, you would take the same number of applications of that target fungicide to build up a given level of resistance to that fungicide. So in that case, alternation of fungicide may not work. Do you agree? So yeah, I think that's a little complicated. I was, I interviewed for a job like 10 years ago at the BASF, and the hiring manager asked me this kind of question. Now he said, if I have a new product, someone told me I can only get 15 applications out of it. Is that right or wrong? No matter what you do it, you only have 15 applications. After 15 applications, that fungicide, the effective life of that fungicide is gone. The fungicide will not be effective anymore due to resistance. Doesn't matter how you do it, you mix, you rotate, do whatever, right? At that time, I said something about fairness cost, but I'm not sure if I convinced him. So I didn't get the job. But anyway, but if there is a fairness cost, I'm just thinking, right? Makes sense. If there is a fairness cost, then in the absence of that fungicide, the sensitive population can take over, can outcompete resistant phenotypes. So you should be able to use the fungicides again, right? Anyway, as I mentioned, we haven't really understood much about fungicide resistance. Now we have to really utilize, leverage the advances in science and technology to better understand fungicide resistance. Now my lab has been working on CRISPR-related technology to better understand fungicide resistance. Now in this case, we use CRISPR genome editing tool to create this specific mutation, E198A mutation in beta-tubulin gene in Coltotrichum siamense. Coltotrichum siamense, that's a pathogen responsible for anthracnose across many fruit and vegetable crops, okay? So the idea is that we create this mutation conferring resistance in isolates with the same genetic background using CRISPR that allows us to really understand the impact of that site mutation, a specific mutation conferring resistance on fairness, right? On fairness. So we can compare the fairness between parental isolates and their offspring mutants. Just have that little mutation, and everything else is the same genetic background, right? So we compare their growth rate, how much offspring that they can produce, and the germination rate. And in addition, we inoculated fruit with both parental isolate and the mutants, and to assess their pathogenicity, aggressiveness, right, on different fruits. And then we're able to track the frequency of wild type, like parental strain in the population. So anyway, all in all, there is no evidence, clear evidence that show there is a fairness cost associated with that specific mutation, which is not good news. But again, I think we need to understand this in order to make science-based resistant management strategies. There are many other mutations, many other pathogens, right? It's just one step, and we have a way to go to really better understand this. Okay, now what are other factors affecting fungicide resistance management? Well, like tank mixture, I briefly touched on that a little bit with a tank mix with a multi-site fungicide. We go with high or low doses, right? So those questions probably you also ask yourself, should we spray before or after a rain? To what extent does resistance matter? I mean, you talk about resistance, but if I only have 1% in my population, if I have 10%, what does it mean, right? In terms of control efficacy. So those are the questions that we had. Then yes, there are some studies that have have a look at those aspects regarding like a mixture, fungicide dose, and everything. So Well, you may wonder how fungicide dose is going to affect the resistance selection, fungicide resistance management. It's a little complicated, but let's say two different scenarios again, right? Between high dose and a low dose, right? If you refer to a fungicide label, it gives you a range. I mean, I'm not saying that's a high dose or low dose, the same high dose, low dose here, but you have a flexibility to go with a lower dose, higher dose, right? Now let's say with a low dose, what does it do? Well, again, this pool, most of your individuals are sensitive. With a low dose, you're going to eliminate some sensitive individuals, but not all, right? But with a high dose, you're going to eliminate more sensitive individuals compared to low dose. Would you agree? Right? Because the dose is stronger, it's going to kill more. But however, it also makes sense that with a high dose, it clears the way for resistant individual, that black spot, to invade, to occupy the area, to occupy the tissue, right? And with high dose, it reduces competition between sensitive and the resistant individuals, so that resistant individuals could take over more easily compared to low dose scenario, right? Okay, now I had a graduate student who was investigating the efficacy with all those questions, factors that we had in mind. So I had a graduate student looking at the efficacy of fungicide spray tactics for botrytis scenario, and factors that are considered in this experiment, including initial resistance ratio. Like I said, to what extent resistance really matter, 1%, 10%, 30%, right? And pre-post infection. That's kind of somewhat related to like a spray before or after rain. Not the same, but it's the same idea, right? Should we apply fungicide after the infection has occurred, or should we apply fungicide prior to infections, right? And fungicide doses. Now, with those questions, we did a detached fruit assays in the lab. Basically, we inoculate fruits with botrytis spores, like resistant and mixed with resistant and sensitive individuals, and we spray fungicide either before or after the inoculations. And then we keep tracking of the development of the disease lesions, and then re-isolate the population and determine the frequency of resistance under different treatments, okay? And the measurements including disease control efficacy. We cannot just talk about fungicide resistance management without talking control efficacy. These two have to be considered together, right? And this figure shows you the fungicide efficacy under varied frequencies of resistance to fungicide. Fungicide, that's a group 17 fungicide, which was an excellent fungicide for botrytis, but now we have some resistance issues. Now, anyway, the x-axis, that's the average resistance rate, resistance frequency, and then the y-axis, that's average disease control efficacy. Now, if we only had a little bit resistance, let's say there was no resistant individuals to begin with, then your control efficacy is almost 100%, 90%, right? But once we start to introduce only 10% resistant individuals in the population, your control efficacy dropped about 20%, or more than 20%, actually 30%, right around 70% here, right? And that's when you do not mix with captain. The first figure to the left, that's when you don't have a captain. You just apply fungicide by itself, okay? But when you have when you tank mix with captain, tank mix fungicide with captain, you can say that with a mixture of captain, it helps retain some of the control efficacy, and also helps a little bit with resistance management, too. And those two figures basically shows the effect of fungicide mixture and the timing on disease severity and the resistance selection. Now, the first figure, again, that's somewhat similar to the previous figure, where it shows to what extent the lesion the disease control efficacy improved and the resistance rate management improved with a mixture of captan. You can see here, with captan, you got less disease lesion, and you got less resistant population, resistant individuals in the population. So there is a clear benefit when you tank mix with multi-site fungicide like captan. Now the figure to the right, that's the timing. We spray before the infection or after the infection. Now it's interesting when you do not have when you compare with captan and without captan, right? And with regard to the control efficacy, there is no statistical difference. But if you're talking about fungicide like resistance management, fungicide resistance selection, the here it doesn't really show clearly, but the pre-infection, so you apply fungicide, apply the mixture prior to the infection, resulted in actually higher resistant individuals. So in this case, you spray fungicide after the infection ended up resulting better resistance management. That's what I'm saying. So, which is kind of different from what we were told. Go ahead.
So would you correlate that to you're better off to spray after the rain?
That's a good question. Well, I think the rain is going to initiate infections for sure, but there might be infections have already existed prior to the rain, right? We cannot say that. There are some what so-called latent infections. Without symptoms, you don't really know you have infections or not, right? But yes, the rain will speed up that process. But I cannot say for sure that this correlates with the field condition. So anyway, but it makes sense to me. Think about this. You have, let's say, 1% of resistant individual in your population. Now, if you spray fungicide before infection occurs, then you does not allow the opportunity for the sensitive individuals to infect. Right? Only resistant individuals can infect. Then as a result, you ended up having more resistant individuals. But if we spray after the infection, that allow sensitive population to play a little bit, and then we create that competition between resistant and sensitive individuals so that they make it harder for resistant individuals to compete with sensitive. That's my take. Yeah. Please.
Sensitive and insensitive, do they reproduce at the same rate?
Oh, that's an excellent question. That's why I'm talking about fairness, right? So we don't know. Yeah, there might or may not be fairness cost associated with resistance or what you call insensitivity, okay?
Because I was always told, no matter what you do, there's going to be some there somewhere. And when it rains, it's going to go explode population-wise. Okay.
Yes, because that rains brings in favorable environment conditions for pathogens to take over, right? To take off. That makes sense.
If you've killed off all the ones that you can kill, and you only have the resistant ones hanging around,
correct.
Are they going to somehow reproduce much faster and take over?
That's what I think might happen because you reduced the competition between your sensitive and the resistant individuals. Right? You cleared the way for resistant to take off.
Right, but can they reproduce faster then?
That's related to fairness. If they're able to produce as wild type, as sensitive, then I think yes. But I don't know. That's a fairness cost question again.
So you can't win.
Well,
I mean, that's the gentleman who said, well, you can only use something 15 times. I mean, clearly he was right, only it might not be 15.
If there is no fairness cost, yes, correct. But if there is fairness cost, then in the absence of that fungicide, then the sensitive population will go up, right? And then your resistant population will go down because it cannot compete with sensitive population when there is no fungicide pressure. Right, that's why I think we need to better understand fairness cost. That's just my opinion. Okay, all right, well, I'll get to that maybe with my last slide. Oh, okay, I know this sounds a little complicated, but now I touched based on fungicide dose, right? High dose, low dose. I'm sure many of you like to use high dose, go with high dose to kill it early and kill it hard, right? And to eliminate as much as possible. But from resistance management perspective, I'm not sure. Now, in the experiment, we included multiple fungicide doses of fungicide, and the x-axis, that's different doses of fungicide, right? The highest is the label dose, actually close to 1,200 ppm, parts per million. And then we also included 3 lower, what we call experimental doses. The lowest is 25. You do the math, it's much, much lower than the label dose, right? And then we look at, again, the disease control efficacy, and as well as the resistance management. And the figure above, the upper portion of the figure, that's the disease control efficacy. Now, all in all, there is not much differences in terms of disease control efficacy among those different doses. But when you look at the lower portion of the figure, that is for resistance management, the lower dose, the lowest dose, 25, actually resulted in the best resistance management. Okay, so it's clear that the lower dose helps with resistance management better than high doses. Now, the dose rate has been a debate, actually. There is a paper that says that, does the risk of fungicide resistance increase or decrease with dose, right? I'm going to read here. 7 peer-reviewed and 4 non-peer-reviewed publications report experimental evidence all show increased selection for fungicide resistance with increased fungicide dose, except for 1 peer-reviewed publication that does not detect any selection irrespective of dose, and 1 conference preceding publication which claims evidence for increased selection at a lower dose. Okay? All right. So, all right. Well, I'm going to summarize what I talked, and with a table, not my table, also from a publication, very good publication, and it shows the impact of resistance management tactics on resistance selection. With all that we just discussed, now here are different tactics, different strategies. Increased dose. What do you think? Would that increase selection of resistance, or gonna decrease the selection for resistance? Or there is no effect? And this is all based on experimental evidence. Okay. And only 2 that showed otherwise. There is 1 showed there is no effect. How about increased spray numbers?
I mean, whether at the beginning of the season, or in the middle, or the end?
I don't think that there is collection with that.
Because when you had your chart up there, give you an example with the Captan.
Right.
It didn't give you as much. Without the Captan, you had great coverage in the early part, but then you had much less coverage later in days past. But with the Captan, you had sort of a nominal coverage in the beginning of the spray, but as it went on to the end, it wasn't as great.
Because of rainfall?
It seemed like, I'm wondering if because you started out with a heavy spray in the beginning, and it knocked out a lot of the
initial inoculum?
But then resistance picked up towards the end. But with the Captan, it was a moderate amount, and then as you got to the end of that clip, it was still moderate. It wasn't concentrated with resistance like the one before, not using Captan.
I don't know. I have to
Usually you start out at the beginning of the season with Captan.
Correct.
And then you go with something else. and then you add the Captan, you keep going.
Right?
But yours was, your review there was simply no Captan and then Captan mix.
Yes, that's what we did with our lab experiments. Correct. But yes, early season you use Captan, right? You could use Captan, and that would help with resistance management.
Right.
And then you think that the resistant individuals, you help kind of control that resistant individuals with Captan applications early in the season. Is that right?
Yeah, you had more resistance management in the beginning with a product, but when you tank added Captan compared to,
Now what do you do?
didn't have as much, but you didn't have as much resistance later either.
How do you know that?
I was looking at your chart.
No, my chart doesn't really reflect that. Which chart? This one?
No, back.
Oh, back, yeah.
There you are. Whoop.
There you go, go back.
Okay, this one?
Yeah.
All right, well, this one is not really a seasonal timing. I mean, it just shows the...
Do you see the concentration of resistance later on?
Correct. That's when you have a more resistant individuals,
Yes.
right? And then that really decreased the control efficacy.
But with the Captan, it was a moderate all the way through.
Yeah, that's the whole point, is Captan that helps with resistance management. Because Captan does not select
So
for
that's why I was
resistance.
asking you if the study had done what the difference is in what part of the season it's in.
No, this has nothing to do with seasons. This has nothing to do with
So
seasons.
nothing correct.
So let me explain how we did this experiment. Okay, so we make those inoculant with different ratios between resistant and sensitive individuals. And we inoculate those inoculant that we made with different ratios to the fruit. And we measure them individually, separately. And then we combine everything to make this chart. That does not have anything to do with season, with timing.
Yeah, that's a problem. That's right.
There is no time, but I get you what you're saying. From my perspective, I can try to answer this question. With Captan, it's like water. Let's say it's not like water, but it does not select for resistance, right? But you want to really decrease the resistant individuals. If there is no fairness cost, the resistant individuals will always be there. They will not go away. You will still maintain the same ratio, right? 10% resistant individuals there in the initial population. With Captan, in the end, you still have about 10% resistant individual if the resistant phenotypes do not have fairness cost. Okay. It just doesn't select for that. Yeah.
Is 10% an economical cost you can live with?
Yeah, so that's why I'm saying here, if there is a 10%, right? If we do not mix Captan, 10% here, that your efficacy would be compromised by 40%, 30 to 40%. In this case, based on our data. Right, so you can say the y-axis, that's a control efficacy. When we do not have any resistant individual, it's almost 100% control efficacy. But when we had about 10% resistant individual in there, the control efficacy dropped to right around 70%. Okay, all right, well, let me finish. I don't want to take too much time. But anyway, so I'll just say increase spray numbers. Remember the figure that I showed in the beginning? It's a selection process, right? Fungicide is your selecting agent. Obviously, if you have more sprays, you're gonna spray for more resistance, okay? And how about split dose? Split dose would also result in selection for resistance because you split dose, but meanwhile you increase the spray numbers, okay? And add a mixture partner, like do tank mixture, right? This is, I think, easy, helps resistance, it decreases selection. How about alternation of or rotation of fungicides with different modes of action? Would that help? Yes, correct. Okay, just timing. Now that gentleman just said timing.
Yeah.
Do you think that would help, or there is? That's pretty inconclusive. There is not much evidence. Yeah. Yeah, it's hard to handle. Environment condition, I mean, everything is just. Yeah. So anyway, now to summarize, okay. I think the best fungicides are not as good weather conditions. If we do not spray, then we do not select for resistance. The less is more for resistance management. Okay, so we wanted to minimize our unnecessary sprays for fungicides that have high risk for resistance development, like those single-site fungicides. And we only spray as we need, when we absolutely need. And then primarily use multi-site fungicide for controlling pathogens with high risk. Like multi-site fungicides, again, they're not prone to resistance development. You wanna use them more to better control fungicide resistance, right? Perhaps use a mixture of a low-risk fungicide. Low-risk fungicide here means low risk for resistance development. Now, low-risk fungicide at a high dose, and a high-risk fungicide at a low dose when the disease pressure is high, right? And again, wanted to limit the frag groups to maybe 2 applications per season, each frag code. Anyway, I think that's my last slide. Unless you have more questions, I'd be happy to answer. Try to answer. Thank you. Yeah.
So if you know you have resistance out there, and you want to try to get the resistant types to breed with the non-resistant types to hopefully get
reverse.
Reverse it. Is there any strategy for that, or is that?
Yeah, that's why we're trying to understand the fairness cost. So if there is a fairness cost, so in the absence of that fungicide, then the resistant individuals will gradually disappear because it cannot compete with wild type in the absence of that fungicide. But in the presence of that fungicide, yes, with regardless of fairness cost, the resistant population will be selected much faster. So.
I remember when Bayleton first came out, and it was a miracle. I sprayed on my pumpkins. I thought I was going to give up on them, and it was an absolute miracle. Next day, they looked like the brightest thing in the world. But that was short-lived. So Bayleton never came back to the market. Would Bayleton work now? I guess that's what I'm wondering. Or because we have so many, what is that group for steroid inhibitors out there, it wouldn't make any difference.
Yeah, so it's like, that's a good question. I think it makes sense, I think, to start really detect for resistance to our old chemistry, like those first generation single-site fungicides. Many of them have not been used for 10, 15 years. And say if we, what's the frequency, better understand the frequency of resistance. Say if there are any, maybe we can use them again, right? While we're losing some fungicides, maybe we can start using the old ones that we haven't used them for a while. Now this slide, we start this monitoring program 2011 to 12. Now the first fungicide I just want to show you, that's a cyphenomethyl, Topsin M, right? Topsin M at that time was used frequently for botrytis management 10, 12 years ago, right? And then we found a resistance to Topsin M, about 70%. That's the time we asked growers stop using Topsin M. It's not gonna do anything for you. And then growers participated in the program, they follow our recommendations. But still after 4 or 5 years, there is no Topsin M. The resistance is still very stubborn. The high frequency of resistance still remain in that population, in botrytis population, right? So I don't know how long it's going to take for the frequency to drop. Hopefully there is a fairness cost, but we will see. But some of them, some of the pathogen resistance, I think they might have some fairness cost. So anyway, yeah. Any other questions? All right, thank you very much.
share your survey for me. There's two surveys in there.